We will get started. Dr. Mani?
Yeah.
Yeah. I think we should get started with this session. Thank you for chairing this session.
Sure. Yeah. Thank you. Yeah. The next speaker, we have the session on entrepreneurship journey with MPS models. And Subhadra, actually we all refer to her as Subha, and she's a PhD from University of Boston, somewhere in the US, and then she did a lot of postdoctoral research in stem cell research. She came back to India and she became an entrepreneur. She's an expert in stem cell biology. In fact, she has biobanking services also. She's been spending a lot of time on developing druggable formulations for several therapeutic areas. And more importantly, she's actually recently working on microphysiological platforms as well. And what she's going to be talking today is on vaccine safety and efficacy testing on humanized MPS models. Welcome on board, Subhadra. We'd like to hear what your take on MPS models. Thank you.
Yeah. Thank you, Mani.
Yeah.
I'm audible, right? Yeah. All right. And I want to thank the organizers for this very interesting opportunity. And I see some familiar faces also, because some point of my previous life, it's like BC and AC. I was part of CCMB but for a very brief period. I was with Dr. Lalji's and Dr. Satish's lab for some time. So I have really fond memories of the building and then the people, very nice smiling faces and all. So I'm happy to be here. So I will be, in this talk, I will be talking about NeuroSafe. This is a solution that we have developed as a non-animal assay system that leverages human stem cell technology, which is microphysiological system. To define it as microphysiological system in this context, and robotic process automation. I'm Subhadra. And in this talk, one of the advanced use cases that we could generate some interesting science-based data from NeuroSafe module is to do with the vaccine safety testing space. So I will bring you all to speed with some background information on, the vaccine safety concerns, and somewhere in between, I would also touch upon what are the traditional methods of assessing vaccine safety and our credibility. In fact, I run a group and which has got some proprietary stem cell technologies and then some products and services as well we have developed. So what is our credibility, and then what has actually brought us to this space, and then I will go about giving some details on the NeuroSafe journey in this talk. I will start with a background, the problem statement. Like anybody else, even in your PhD, you would start with the problem statement and then go about, finding solution for that, right? So for us, we chose this problem statement, which is to do with vaccine safety concerns. So it's actually historical, and since 1955, you will be really kind of surprised to know that in 1955, to 1963, there were reports of polio vaccine causing paralysis in the immunized population. So it was the first incident to acted as an alarm. And then in 1976, the swine flu vaccine seems to have caused a serious neurological disorder, which is Guillain-Barré syndrome, again in the vaccinated population. These are all something would fall under AEFI, adverse event followed by immunization. It's not your preclinical safety data, and then the industry would have some recourse to go back to. But this is right in the clinics and then in the immunized population, because see, immunization or vaccination is given to otherwise healthy individuals, right? I mean, they're not patients, but then if there is some safety concern and then some kind of a debilitating disorder caused to the healthy individual otherwise, it's not something, anybody would want to even think or know about. Likewise, in 1998, progressive nerve disease, it's progressive, which was again associated with the hepatitis B vaccine. Then followed by, the Menatra in 2005 and 8. And the last one, of course, before COVID, it was 2009. H1N1 influenza was also associated with something called as narcolepsy. So there is a historical, a concern associated with the vaccines and then the very immunizations itself. Then in our lifetime, the virus, COVID-19, which is a human SARS-CoV-2, has really kind of caused the secondary infections to do with neuropsychiatric complications and all. And again, very well documented research and study was published in, the prestigious The Lancet and then Journal of Neurological Sciences and all. So this is just the hint at the virus being human neurotropic in nature. So I'm bringing to the context of the human neurotropic pathogen having to do with the possibilities of causing something to do with the vaccine safety concerns. So this is to do with virus. Likewise, COVID-19 vaccines also, FDA had given black box warnings to all kinds of, the vaccine producers, not just, the attenuated or inactivated virus type of vaccines, even to do with your Pfizer and Moderna also, they were put on hold. Some of the batches were put on hold because of the concerns that were found in the immunized population. So it's actually, there is a real-time evidence. History is, of course, we were not part of it, but then this COVID-19 period did kind of open up, some of the Pandora boxes vaccine safety concerns, and then again, especially to do with something called as neurovirulence. So it's actually neurotoxicity, but then if a vaccine is causing neurotoxicity, it's called as neurovirulence. It's defined as neurovirulence. So that's just about, genesis and then building the problem statement. Now, WHO is very well aware of, which is, it's not a regulatory body, but then like a big daddy. And then they have set certain guidelines, what are all the kinds of vaccines that have to be tested for neurovirulence, not just during the preclinical stage of development, but also periodically, as long as these vaccinations are in the global immunization programs, periodically the seed stocks and then the vaccine candidates, produced batch-wise, have to be tested for something called as neurovirulence. So, see, I just want to distinguish between your chemistry, to do with the small molecules, pharmaceutical industry that deals with the small molecules, only at the preclinical, because they are well defined, only at the preclinical stage, the group or the team, whoever is working with that particular candidate, have to do, the entire profile of assessing, the safety concerns, and then once it is done and then they get the FDA approval, while the drug is being produced, they don't need to worry about the safety, of course, after completing the clinical trials and all. But anything to do with biopharmaceuticals, and then if there is any kind of a live organism, which is taken as the basis to build the final product, in this context maybe vaccine, as long as these vaccines are will be there in the markets, periodically the safety needs to be assessed in addition to preclinical and clinical stages of development. So of course, like there are rules, and then like your vaccines like polio, measles, mumps, rubella, and dengue. In fact, like dengue also, like, and influenza. So these are the list of vaccines that have to be periodically assessed for the safety concerns. Now, there is this gold standard method of assessing neurovirulence, and which is monkey neurovirulence test. It's called as MNVT, and traditionally a global industry has been practicing this assay methodology for the past 50 plus years or so. But as it appears, these are like big animals, and then it's too cruel, cumbersome. But then, industry did not have any other option or alternative, so they're forced to adopt and practice this MNVT. So even with MNVT, there is this mandate that at the preclinical stage of development, it needs to be tested once, and then the seed stock on which the vaccine candidates are built as well have to be periodically tested. And so that periodicity is also set by the WHO-like bodies. So alternatives to monkeys, different groups globally have been working on rodent models, mice models, transgenic mice, including some in vitro methodologies like MAPREC and all. But then other than MNVT, the rest of the assay methodologies, they're still at a very research stage of development, and then they're yet to get integrated into the industrial application. Then with that background, during COVID, pandemic has actually magnified the issues to do with sourcing animals, and then modeling, using the animal models as important platforms to test safety and all. So you will probably laugh at one of the news item that said there are not enough monkeys running around, and then the country, especially the US, was facing a monkey shortage because very cleverly, the Chinese wildlife ban has frozen export of test monkeys, to the entire globe. So the imbalance was created at, the country like China, while the entire globe was affected. So as I said, there just aren't enough monkeys to go around was the news item in 2020. So it was magnified, and then in this year, 2022, US House passes legislation to end FDA animal testing mandate, and then in 2020, regulatory bodies like FDA, CDER, they have given their perspective, their opinion on looking at or exploring these NAMs, new approach methodology. So what I want to convey here is there is this real problem with the animal models and sourcing of animal models, and then on another side, the regulators are as well, creating some kind of a block, and then they are encouraging the industry to go for alternatives. But then where is the alternative? So they're there. The different groups have been working, but then integration into the industry has been taking its own sweet time. So we kind of sense that. But before that, we also kind of got hold on to Merck publication, which was in 2021. So this was like a very surprise, at the same time, boost in our scheme and then in our thought process. So this Merck, very interestingly, in this review article, they have introduced the concept of exploring human-induced pluripotent stem cell-based platforms as alternatives to these monkey models. So they have compared, what are all the existing methodologies, assay systems, and then at the end, this the review article very nicely kind of discussed, like, the concept of, it was just a concept. They did not have any data at that point of time in 2021. So we picked it as the peer-reviewed concept in our story. Then with that peer-reviewed concept, I just want to introduce to my group. I run this group Transcell, and it has a dedicated vertical named Transtalks Bio, which is all about, human surrogate, in vitro, cell and tissue based platforms. So the vision of this vertical is to offer safety and efficacy testing as a solution to the global industry, while the mission has been to support the industry to adopt cruelty-free operations. So indirectly, like, encouraging them to go for non-animal assay systems or methodologies in their life cycle. So we did not want to restrict, like, hey, you should be probably adopting at your preclinical stage of development, like, certain non-animal methods. Like, we've been, because I represent this group, and which is a private limited company, we want to be very meaningful and then, like, smart in our proposition. So across the stages of development till, the production and manufacturing of pharmaceuticals and biopharmaceuticals, we wanted to offer these new approach methodologies, which are non-animal based, to find spot in the application. So that has been, the history and then the background and then the strength of this particular vertical. So in fact, like, we also, again, before COVID, for at least 3 years or so, we were supporting small global biotechs and then small CROs and then also some academicians with the supply of these cell and tissue based configurations. And in our universe, in Transtalks Bio universe, the source of this biological material to harvest and configure these cells and tissues happens to be biological discards, because we come from, my team comes from, like, the combination of both academia and industry background. So we very well know that, like, if you have to rely upon a particular source, there has to be continuous supply of the source, because if the source, if there is any paucity of the source, your entire, like, system or like whatever the protocol that you have built would go, like, berserk, right? So for example, if skin biopsy is something that, like, you're relying upon to harvest certain primary and progenitor cells, you obviously can't, like, get unlimited supply of skin. Likewise, any other biopsy. So we knew about it, and then, like, we have some experience, because I also run a biobank from Hyderabad. So we have a repository of the biosamples from, of all different kinds. So our repository consists of tumor tissues and also healthy individuals' cord blood and tooth pulp. So that it's kind of a strength in our story. So we could get access to some of the biological discards like cord blood, cord tissue, tooth pulp and bone marrow. So we kept that as them in the labs, and this configuration dictates the use cases in our context. So these configurations, because of these special configurations, and then some of these are even proprietary to us, the platforms are made phenotypically responsive, genotypically reactive and functionally approachable. So this is kind of these three traits of the configurations from Transtalks Bio, is kind of continuous whenever and wherever, like, we are pitching to the potential user. So that is another USP. And then the ease of handling and because, like, we deal with biological samples, and of course in our production labs we produce, like, following the GMP protocols, and then, like, whatever the guidelines that one needs to be following to make sure that even not just the characteristics of these platforms, but also sterilities also, because I was listening to the scientists from in STEM, who said they make sure that they supply the sterile and then the compliant cell lines. So likewise, we also have been following, and at the end, there is absolutely no need for extrapolation of the readouts if any of our in vitro platforms are part of any assay system, because the origin of these cell and tissue based platforms happens to be human biological discards. So that's an interesting angle that we brought to the table. And so I want to introduce here this platform named Trans-MSc. So this is a trade name that we have given to the basket of offerings in our vertical, Transdocs Bio. So this Trans-MSc is a very well characterized human induced pluripotent stem cell platform. And when we harvest this and then produce it in batches, we have our own methodology of inducing pluripotency, and then we don't follow Shinya Yamanaka's protocol. Of course, his protocol is, his protocol got a Nobel Prize, but then this is something that we have indigenously developed, and then our confidence, to do with the characteristics and then the induction of pluripotency also stems from the data that we have generated surrounding this platform, and we have some credibility that we went about publishing it as well. So it's a very well characterized when it comes to the gene expression profiles, as well as the cellular kinetics. So this became the basis in building our NeuroSafe story. This is the cell-based platform that we dealt with. Again, we come from, as I said, we have this experience and then credibility of supplying these cells as platforms to multiple, the users, and some happen to be industry also. We had to generate this certificate of analysis. So this itself speaks that it has gone beyond any small scale or laboratory based preparation of this Trans-MSc. It's in large quantities, and then I think we must have produced more than 1,000 batches of Trans-MSc in our facilities, and so we were able to establish these analytical parameters when it comes to identifying Trans-MSc. So we use this as a cell-based platform, and then we brought, we introduced this digital framework that we have procured from Amazon Web Services. So we procured some of the pieces of the framework, and we have assembled that framework again in-house. And this digital framework also is embedded with certain AI and ML tools. So we kind of learned that there are close to some 8 different complex algorithms to deal with biological data, complex biological data. But then of those 8 which are published, we had to resort to neural networks, because that is something which we have realized that can handle the phenomics data acquired from Trans-MSc, treated Trans-MSc. So I'm bringing the link with Trans-MSc as the cell-based platform, and this digital framework which is trained with the phenotype and genomics data acquired from treated Trans-MSc. So this became solution that we offer with 2 components to a potential user. So for example, if we are giving it to Mani or maybe Dr. Suresh, we supply this trans MSc as units to them, and then we give access to this software through cloud infrastructure. So this software is trained with more than 20,000 single cell imprints acquired from trans MSc configurations. Am I making sense? Yeah? Okay. So this particular software as well has its own, the parameters that it has to comply, and then it is so exclusive. It is built in such a way that it is exclusive to trans MSc. So this software will work only if somebody is using trans MSc as the cell-based platform, but not the other way. Trans MSc applications are more than one, but then all I want to say is this software and trans MSc are not mutually exclusive, but then this software is exclusive to trans MSc acquired phenotype and genotype data. So features like, it's very modular. This particular software is modular and flexible. Like for example, today, we deal with some global players like GSK and J&J on another side. So it nicely fits into GSK's framework, as well as it's built like a modular framework, so it will also fit into J&J's framework. So that's the kind of flexibility that we have designed into this solution. So we believe that it is a revolutionary product offering with inbuilt bandwidth to be integrated in the user's workflow owing to the robotic process automation that is part of this solution. And we were very fortunate to get the entire protocol and then the assay system as well reviewed by ICC-VAM. So this is, I'm sure whoever is part of this 3 R's strategy in animal testing, would know this committee, which is NIH accredited and all. So they're like, how do I say, they're a non-profit agency, but they support and they review the data and then the protocols developed like non-animal methodologies and all. So that is the credibility when we speak and then when we go and offer it as a solution to the potential user. So as you see that, it leverages a human microphysiological system as a model for evaluating certain safety and efficacy concerns of biopharmaceuticals. So we have developed it as next generation workstation solutions that combines these cell tissue configurations with image and data analytics, but seamlessly integrated in the user's workflow. So see, it is actually the way we offer this solution to the user is antithesis to the traditional CRO models. In CRO models, the sponsor, who is the industry, needs to send out their material to the contract research or contract testing services where this particular testing is undertaken. But in our strategy and methodology, for example, NeuroSafe, we take NeuroSafe to the to the user's facility, and they don't need to send the material out because the way it is built is to be seamlessly integrated in their workflow. So we believe that it is totally antithesis, and there are advantages. Like for example, if it is to do with vaccine industry only, there are several regulations, and then they obviously can't share their seed stocks or wild type pathogens that they deal with in building the vaccine candidates. So it nicely fits into such a requirement where they don't need to send anything out from their facility, and then we take this solution to their facility and then integrate into their systems. And so while we were developing NeuroSafe, we had to deal with not just the real-time platform handling, but also the digital analytics, like sourcing the data from trans-MSC, and then data ingestion, annotation, and aggregation. So when we were developing, we were doing all these steps manually. So training the machine, which is the software, the digital framework, with the thousands and thousands of data sets that are generated on trans-MSC, is very tedious. But then we had to do it manually. But when the solution is offered to the user, this is all kind of made automatic. So it's kind of fortified with the robotic process automation. So at the user site, I'll explain the scheme, how it works. I will anyways go back to a few of those slides, but then I'm just kind of jumping to build the context. Because I want you to appreciate so this is the scheme. So you have this test material, and we supply this trans-MSC as units. For example, in a month, a user can ask for, let's say, maybe 100 trans-MSC units, and we would supply it, and then the logistics of it is very well established. So globally, anywhere in the world, these trans-MSC units can be shipped in the cryo-frozen format. Yeah, so in their microbiology labs, or maybe quality control assessing labs, they just have to treat their test material, with the trans-MSC and incubate it for a specified period of time, and then collect the images under the phase contrast microscope at 20x magnification. So these images would be fed into...
Subhadra, you have 5 minutes, okay?
I have only 5 minutes?
Yeah,
Okay.
30 minutes finished,
Okay.
5 more minutes,
Sure.
okay? Thank you.
Sure. Yeah, so these images would be, at least a minimum of 20 images would be fed into the software, the access of which would be given through cloud to the user, and then within 5 to 7 minutes, the score would be published, as a report. So where is this 5 to 7 minutes? And then if one has to compare monkey neurovirulence test, you will probably laugh at the time period that is taken for completing, assessing some of the safety profiles like neurovirulence in monkey models for at least 5 to 7 months. So 5 to 7 months is traditionally the time taken, if one is practicing MNVT, but then if one adopts NeuroSafe, it's not even 5 to 7 minutes that they get to know, the concerns like neurotoxicity or neurovirulence pertinent to the test material that they are using. So it's so robust and rapid. Yeah, so it's rapid and robust when it comes to the assay system, and product specific and process specific also. So while we were developing this solution, we have generated the foundational and first set of benchmark panels. Also, when we implement it at the user site, we develop the second set of benchmark patterns on the product that they have chosen. So that would be as well trained in the system that they would be using. So it's product and process specific, ultimately making it very robust, at the same time with an intention to eventually eliminate the need for animal testing. So as you see, it implements Thiar's method, and not just in the preclinical stage, but even post licensing also. There are some, I think minimum of some 100 batches of vaccines produced in the user's facility, have to be as well tested on I think some hundreds and thousands of animals. So our NeuroSafe can nicely find a spot at all the stages of the life cycle of development. So we have a US patent on the very assay system, and we follow when we developed this software, we kind of got inspired with the scoring method that is set by WHO. So the grading as well is the pattern is taken from the WHO norm, but we have kind of customized it to the kind of cells that we are using. So as you see, there is this integration of data from wet lab, which is passed through the trained software, and then the readouts on human neurovirulence to do with the signals that are picked from the treated trans MSC is the story of assay system. Maybe I think I'm good.
One more minute.
Oh, I do have? Okay.
Yeah.
I'm going backwards. So we developed it in such a way that this assay system is even suitable in detecting microbial contaminants and then adventitious agents in the product, in addition to residual neurotoxicity and neurovirulence. At the same time, we also have partnered with one IT giant, where we are offering and extending NeuroSafe's application in detecting neurovirulence signals in pharmacovigilance also, which is to do with the clinical safety assessments, while majority of the features of NeuroSafe is to do with non-clinical safety assessments. Yeah? This slide basically refers to the compatibility of the platform, which is trans MSC, to do with small molecules, biomolecules, and of course vaccine candidates is the use case that I have taken you through, and then CAR T cell therapy also.
Your time is over.
Yeah, sure. I'm sure you all must be reading on the CAR T cell therapy causing big time neurotoxicity and all. So we are kind of we have started pitching to some of the big CAR T cell producers, in the globe.
Thank you, Subhadra. Very wonderful presentation. Only one question for one person, maybe 3-4 questions, but don't ask multiple questions. If you have multiple questions, you can take it outside. Okay? We have a few question time. Please go to the microphone and ask your questions. Thank you.
Hi, thank you. That was a great talk, a great product. Good example of how we can combine AI and data science and biology. So I have a question regarding the algorithm. So you've used convolutional neural networks and probably some deep learning as well. So generally, when we use these models, we don't really know what's happening inside in terms of the statistics and how the model is predicting the results. Has this been an issue for you when it comes to, say, regulatory or things like that? Because for clinical applications, they may want to know what's happening inside the algorithm, right?
Your question is based on some of the existing regulatory permissions that we have received, compliances as well, based on the compliances. So the framework did not kind of have any issues when we started with, but then these guys were not keen to know, how the complexity is addressed within the algorithms. No, because they are only looking at the readouts and then the biological significance of the assay system that we have developed.
Okay. Huh? Yeah. Thank you.
Good. Anybody else? Students? Subhadra has a lot of job opportunities. Please get your attention right now. Okay. Anybody else? Right. Subhadra, I do have one question. It's a general question. You're talking about a NeuroSafe vaccine. What is the incidence, a percent incidence of these, whether the side effects of this polio vaccine or Guillain-Barré syndrome, what are the percent incidence and population. So, for example, somebody got a polio vaccine.
Yeah.
What is the percent of incidence?
Right. See, as part of every company's pharmacovigilance, I mean, they have to be, there is a mandate to be collecting, like, the data forever. Yeah. So there are incidents, because I went to that database, like, just for my knowledge in terms of, like, what is the percentage of incidents for polio and all. Last year, I think, like, I mean, there was one small report which was published, which speaks on, again, like, I mean, polio vaccine cost AEFI, adverse event. Polio is one, at the same time, measles also. Also, yeah. So there are, of course, in India, we do not have any published data, because we are not sure of, like, I mean, whether they are doing the pharmacovigilance or not. But yes, there are incidents of happening. Even if it is like less than 0.1% also, it's a big deal. Like, I mean, it's not supposed to cause any, adverse event in the immunized population. Yeah.
Yeah. So, since we are actually talking about this as an entrepreneurial journey talk, what do you give advice from your entrepreneurial experience to the students or postdocs here who are listening? Just give it 2 or 3 minutes.
Right. No. Do you really want to know? I mean, postdocs and PhD students.
Okay.
So, like I was wondering about the validation of the product. So,
Right.
When you go to the regulatory agency, they say you at least test on, say, 100, 300, 500. So that was one. And when I was curious when you said we take the product to the company.
Right.
So I was curious how big the product is and how does it look. And so maybe one suggestion is whether instead of these many slides, just one video, 3 to 5 minutes video on how the product. Video on how the this NeuroSafe actually. How does it work actually? Some small 2 to 3 minutes video would definitely capture the audience. That was a suggestion. Thank you.
I do not have time, but then I will share it with you or with anybody, whoever is interested. Yes, for your question on, I mean, we take it to the user, and then, like, how does it work in terms of the data? So, Bharadwaj, that's a good question. While we were developing, and of course, I've been part of this, the development, commercialization, and of course, I've been on both sides, like, one who was using it when I was doing my PhD, and then one who has now developed it, and then I want to kind of support, the industry. So there is no end to the kind of expectation in terms of, like, I mean, how many data sets, or, like, what is the level of validation of the solution, that there's no end to it. I knew it, okay? So the way we kind of offer it is, we want the user to adopt it fast and then choose their product of interest, because 100% we would not have had access to their product. For example, if it is vaccine industry as such, there's no way you can get, maybe we can get some mimics outside, like, I mean, commercially available candidates and then the compositions, but then it's impossible to get their finished product. So when we implement it, we have this first acclimatization and customization period where it would go up to 3 to 4 months, where we generate the user's product specific signals and patterns as the second set of benchmark patterns in the NeuroSafe solution. So that would answer all their concerns, because when we present data pertinent to their product, that's enough for them, and they don't need to kind of ask for any other validations, because while we customize it, we establish standard curves and ranges specific to their product's composition. Yeah?
Thank you.
Images of trans MSC. Yeah? So from the images, the software picks the percentage of affected cells, and affected cells could be necrotic cells or apoptotic cells. we have trained it, or even cells in shock. Yeah, so there is a score, like, that goes about enumerating these, and then on top of it, like, some cells with the infiltrations also. So there is a formula that we have built. Again, as I said, like, the scoring method that we kind of got inspired is from WHO, that is set for MNVT. Yeah, so there is a scoring method inside, and then ultimately, like, it reports the residual neurotoxicity or neurovirulence signals. Yeah. Okay.
We have just another 2 minutes for you if you want to talk about your entrepreneurial experience in just 2 minutes.
Sure. Dr. Radha said we have this next session.
I am tracking the time.
So, imagination, don't limit, because like when you are working in the labs, because you use your hands, and then, like, you use your eyes and you use your brain, you very well know, like, what are the limitations and all. But then, if you want to translate your lab data to certain applications, limitations would be there, but then, like, you have to see how you would fill up the gaps, and then, like, take it to the next level of translation, only if you are interested to go in that route. That's what, like, if that is what is the mindset. So be bold. I mean, it's okay even if you kind of fail. Let's say you fail some 10 times, but then, at least I was very hopeful. If your hypothesis is right, and if your protocol, obviously owing to all your experience working in the labs, would be right. So at the end of the day, you need to pursue, and I'm sure, like, you will see the light at the end of the dark tunnel, which is entrepreneurship. Am I right? Yep. Suresh? Yeah.
Thank you. Subhadra is going to be available at tea time. Anybody has any questions can consult her. Okay, thank you.